Okay, this is part 3 in our 4-video series of predicting genes in a genomic DNA sequence. In this video, we will learn how genes are identified within a FASTA sequence based on codon usage and coding potential. There are 4 types of evidence that may indicate the presence of a protein coding open reading frame in a phage genome. First, an open reading frame that is long enough to encode a protein of at least 26 amino acids, thus an open reading frame that is greater than or equal to 81 base pairs. Second, the presence of a Shine-Dalgarno sequence. Third, the presence of coding potential. And fourth, high-quality protein sequence alignments to known bacterial or bacteriophage genes. We have already discussed open reading frames and Shine-Dalgarno sequences, and we will talk later about how we detect Shine-Dalgarno sequences. We will also discuss later how we use the bioinformatic tools such as BLAST-P or HH-Pred to match putative gene-protein sequences with known protein sequences and databases. In this module, we will focus on what coding potential is and how it is detected. Coding potential is the probability that a DNA sequence codes for a protein. This graphic shows the probability on the y-axis that a given sequence on the x-axis codes for a protein. The probability is calculated for all 6 reading frames of a sequence. The black bars indicate predicted genes, and each black bar is associated with high coding potential. We will talk more about how to interpret coding potential output later in this module. We use 2 bioinformatic tools to measure coding potential of sequences: Glimmer and GeneMark. Open reading frames that code for proteins tend to have sequence patterns that don't exist in sequences that do not code for proteins. Glimmer and GeneMark programs detect these sequence patterns. We will talk about why protein coding sequences have these detectable patterns first, and then we will talk about how the Glimmer and GeneMark programs first learn what these sequence patterns are, and then how these programs detect the patterns in the phage genomes. First, let's learn why sequence patterns are created in protein coding open reading frames. The main reason that patterns are created in protein coding sequences is that the genetic code is degenerate. That means that there are multiple codon sequences that code for the same amino acid. For example, there are 6 different codon sequences that code for the amino acid arginine. Organisms and viruses, however, often do not use each codon equally, and their preference for one codon sequence over another has to do with the nucleotide makeup of their genomes, or %GC content. The number of C's and G's in a sequence or genome can be calculated by determining the frequency of each nucleotide in a sequence and then calculating the %GC content across the sequence. This sequence has 46% GC content. The genomes of organisms and viruses are often skewed in their nucleotide makeup. The GC content of E. coli is just over 50%. The phages that infect E. coli have similar GC content, just under 50%. Mycobacteria and mycobacteriophages have genomes with high GC content, which means there are more C's and G's than there are A and T nucleotides. M. smegmatis has a GC content of 67%, and the phages that infect M. smegmatis, on average, have GC content of around 65%. Codon usage is the frequency of certain codons used over other codons for the same amino acid by an organism or virus. An organism's GC content affects the codon usage and creates a bias for certain codons over other codons for the same amino acid. If the GC content of Mycobacterium tuberculosis is 65%, which of these 6 codons for arginine will it likely prefer? This is a codon usage table for M. tuberculosis. It presents the average relative synonymous codon usage, or RSCU, which is the number of times a codon appears in a gene divided by the number of expected occurrences under equal codon usage. Let's zoom into the arginine codons. The arginine codons that consist of C's and G's only have the highest codon usage at 2.64 and 1.95. In contrast, codons that are richer in A's and U's are rarely used, with frequencies of 0.21 and 0.04. In summary, in regions of the genome that do not code for protein, such as intergenic regions, 3 base pair code will occur randomly and equivalently. But in open reading frames that code for proteins, the preference for certain codons over others will create sequence patterns that can be detected as coding potential by programs such as Glimmer or GeneMark.